Yeah, so welcome everybody. I think people might be dropping in still. I heard from David, people are still mingling on the Mingler. I didn't know that this was the first workshop, so that's exciting. Yeah, so I'm part of the Bio Art and Design organization team of the Bio Summit. And one of the things that the Bio Art and Design team has done is create an online exhibition, because of course we do all of this stuff online now, which is called the Bio Creation Station. I don't know if you all have seen it already. People been already to the exhibition? Have you checked it? Raise your hands. Not a lot of hands yet, which is also why we do this kind of organized tours to lure people into checking the exhibition. And yeah, it's very cool. Thanks, Tim. And what we do is we organize one every day, one tour with different artists present. So they are there now and they can talk a bit about their project. And you can also ask them questions. So this is all really informal, and everybody should also be able to unmute themselves. So imagine you're in an exhibition and we're walking through this room, right, together. And instead of stopping at all the works, we stop at a couple and just look at it and ask questions about the project or the process. I am going to take you there now. I have to share my screen. Share screen one, share. This is the website of the Bio Creation Station. You can find it on the, you see this screen, right? I'm sharing the right screen, yeah. You can find it on the Bio Summit Live site. Sorry, something happened to my Zoom, which I have to fix. Well, okay. Yeah, so this is the Bio Creation Station. So it's a place to showcase the creative, funky, inspiring projects. And it's been really great. I want to give some credit to the people who did a lot of work building it. Karolina did a lot of work, Karolina Sulic, filling it up, and Heidi Jakkel as well. And so they did great work with people sharing their projects and putting them on the platform. With that being said, so we're going to look at a couple of these works today. If there's other artists present, please let it know in the chat as well. Maybe we have a bit time to talk with you as well if we have more time. In total, we have like 40 more minutes and then we're kicked out the room. So for this tour, I wanted to talk specifically about projects which are kind of a passion of mine, which integrate biology and digital systems, or living media interfaces, they're called sometimes as well, so that you integrate microorganisms in a computer system, or kind of work on the edge between information systems and, for example, DNA. And I think that's a very exciting field. So I try to find people that have something to do with that today. And the first one is Tim Dobbs. So I'll scroll to your project. Imagine we're walking, right? This is a room. And your project is Tim Dobbs.
At the end of the hall.
Huh?
There we go, yeah.
I
said the art is at the end of the hall. We're walking.
The end of the hall, yeah. Oh yeah, more stuff is adding as we speak as well. I see this one is new edit. So if you added something to yourself to the Drive folder, but it's not up there yet, it can still be added in the next days, I think. So this is also a growing collection of projects. Yeah, so let's talk a bit about Bioartbot. I'm opening it now. Yeah, so every exhibition has kind of a big picture there, and then a small text about the project, a bio, and then more pictures at the bottom, which you can click through. Maybe I'll keep it on here. Maybe Tim, take it away. Can you kind of tell a couple of minutes about what Bioartbot is and what we're looking at?
Sure. Yeah. Thank you, Roland. Bioartbot is a platform that we started building last year at Counterculture Labs. and actually kind of soft launched at the Bio Summit last year. So I know a lot of the Bio Summit family people submitted to it. But basically, it's a website, bioartbot.org, where you can make a drawing. And then we use a liquid handling robot and some GMO E. coli that have been engineered to produce different colors, different proteins, to turn your drawing that you made on the website into an agar art piece. So we kind of started this up last year. COVID really threw a wrench in the works, as it has for many people, I think. But we've kind of piloted this, and I just picked out a few of the standouts to me. What I really loved about this project was there were a lot of technical challenges to solve. You know, the website, the robotics, growing the cultures, kind of making that all work. But in the end, it became this very fun tool to use. And I often didn't really even know what the submissions would be. So it was kind of a tapping the creativity of the whole world. And so it was really fun to watch the robot, you know, put down these colonies of E. coli, but I have no idea what the art was going to be, and then watch it grow over a couple of days in the incubator, and it turns out into being something. So I picked five ones I really liked, and I think I'll just talk about them. This first one is it's just a bunny rabbit, and I just think it came out really good. It's very cute, and I really like the sort of pixel style that we end up with, because each little pixel is a colony. Do you want to
Wait,
just click?
what are we seeing here? So this is a petri dish with agar, and you use an OpenTrons liquid handling robot, or
Yeah, yeah, that's right. So yeah, so we bought a bunch of these square agar dishes, and then we just use an OpenTrons liquid handling robot to we grew up 5 different cultures, so 5 cultures of different strains of E. coli that produce different colors. And then the OpenTrons, what happens when we go to print these, we load up, you can fit 9 of them onto the OpenTrons deck. And so we'll do 9 at a time. And the OpenTrons will go and grab a little bit of, say, this blue strain. I'm not remembering the exact plasmids that produces blue. But it goes and gets this blue strain, and it just touches each of the points that the artist sort of programmed into it. And then once it's touched each of those, that's kind of like inoculating each part. And yeah, and Patrick says in the chat that these are the same strains that were distributed to some of the folks at Bio Summit last year. So these are hopefully some strains that a lot of people have around the world. They're sort of just built to have these reporters in them, so they have different chromophores and fluorophores. Yeah, so what happened was this was a blank agar plate, and then we touched each spot with each of the right colors, and then over a couple of days, incubation, then they grew up, and we got this cool picture of a bunny rabbit. And then yeah, we took a photo, and you can see there's a little bit of a black light to try to get some glow out of. There's the kind of glowing one is GFP, green fluorescent protein, and yeah, and then we sent these pictures out. I actually just managed to send a bunch of these out last night. It's kind of still a manual process.
Do you remember who made this one?
Yeah, it was a guy named Ben Tippett. He's actually, he lives in Canada. And he's kind of a physics nerd, which was cool to get to know that, because I think the biology side was kind of gross for him, because he's more into, you know, the physics side of things. But yeah, that's what I can tell you about this one. It was a guy named Ben, and I know he loves bunnies, so that's how he made that one. Yeah, I can.
So, wow.
Yeah, this one, I made this one. It was sort of a test, but I like the way it came out. It's a little lion, and I like the way the glow works here. And as I was thinking about it, I thought it was kind of cool to represent a big scary animal with a tiny animal. Well, not an animal, but a tiny organism.
Cool.
Yeah.
And so this is GFP green fluorescent protein, RFP, and blue fluorescent. These are all fluorescent proteins, right?
No, actually, they're not all fluorescent. The blue one is does not fluoresce. I'm blanking on it, but I can send those out if anyone is really interested. I can dig up. They're all from the they have Biobricks pages. This one I thought was cute. It's you know how you might carve into a tree. You know, K loves tea. And I thought this was kind of a modern update to that. So we can you know, for scientists and bio nerds, we can carve our love into agar plates.
Great. You also have some infections here, I see.
Yes, yes, it was a bit of a challenge in our space in counterculture labs, but I actually sort of liked the way they came out. Like this one, I like the contamination of this bit of mold up here because it kind of adds sort of a spooky feel to it. This one, I asked for, I reached out to the person who submitted this and asked for a sort of artist statement, and he said, oh gosh, I'm not going to get the quote right, but basically his feeling, he was trying to talk about surveillance capitalism and how a lot of our society these days is based around sort of people or companies trying to, you know, see into every aspect of our lives. And he liked the idea of sort of using this tele art system to then comment on the connection. So I thought that was cool. I think it's supposed to be like a sort of an all-seeing eye kind of thing.
You got some, maybe let's do some interactive stuff. You got some questions already. Neville says, mine turned out great. She got them in the mail this morning.
Oh, awesome. I'm glad you got it now.
And Judy Nguyen asks, will you submit these to the American Society for Microbiology agar art competition or make new ones using this method?
Yeah, great question. We submitted exactly one last year. I just kind of couldn't get them made fast enough. And it was a finalist in the amateur section, which made me feel really good. And so, yeah, I'd like to be able to submit more of these. Either, yeah, if we can get new ones made, it might be a little tight this year. But I think some of these we can actually also still submit because they were never submitted and they were made, you know, in the past year. So,
Cool.
but yeah, it's a great idea, and I think they're neat. Yeah, and this last one is the joke was to, instead of Bitcoin, you could make a Bitcoin, which is based on the idea of a lot of computers doing calculations to generate this value. You could say, well, E. coli can generate the value because they're just doing, you know, it's a bunch of E. coli working away. And so you could make a cryptocurrency out of that called Sasquatch. So that was someone's pitch. So invest today if you're interested in Sasquatch. So I thought that one came out neat too. And much more energy efficient, exactly. Catherine. I'm telling you, it's the future. Like, you know, we'll be opening our organic synthetic biology Bitcoin any day now.
So maybe some last questions for Tim before we move on to the next artist. You can also unmute yourself, people. You don't have to type. You have the power to talk. Okay, I have some last one. So this Artbot project, is the website still live, and can people still add to it? Because you said you kind of did the last one yesterday night or so, you said. So this is still something that is live and happening.
Yeah, it's still live. Bioartbot.org, like I said. There's a note on it right now that says you probably shouldn't expect to get your photo back that quickly because I haven't been to the lab at Counterculture Labs in some time. But I would encourage you to make a submission. I think we'd love to print it and get a new batch going because it's really, really cool to just sort of see what comes out. And I really like it as sort of a collaborative art project. The way I wrote it in our emails is that it's a collaboration between you, like whoever submits, Counterculture Labs, and billions of E. coli cells. And we all make art together. So yeah, the website's still live. It's an active project. And in fact, we're trying to grow it. So if you want to collaborate in any way, reach out to me or, go through the website. I think it would be really cool to have this be even, more of a global collaboration because it's sort of built around that concept already.
Well, that's great, Tim. Thanks. Let's give Tim a
Thanks,
hand.
everyone.
Maybe you can share the links one more. I mean, it's here on the website, but I'm gonna. But you can also share it in the chat.
Yeah, I'll drop it in the chat.
Boom, so people can see more info later. And of course, we can also continue these conversations on the Slack after this tour.
Yeah, come find me anywhere.
Yeah. All right. Next artist I would like to focus here is Karolina Sulic. Karolina, are you here yet? Yes, you are.
Yes. Yes. Hi.
In beautiful tropical Poland.
Yeah, Tim inspired me. He's also like in some tropical place.
As I said earlier, Karolina also put a lot of work in actually uploading all this projects here in the Squarespace website, in the Bio Creation Station. So I think it's fair that you can also talk a bit about your own work. This is it, DNA signature. Yeah, maybe you can first tell us a little bit about it, like a couple of minutes, and then we can do some questions.
Yeah, sure. Will I be able also to share my screen?
No, I'm sharing the screen. You're working with me.
Okay. Okay. So this is a very simple, fun project that I made last year, and it was done actually during, I think, working on some bigger project. And it was just playing with coding and DNA, the work that has already been done by many, and I didn't figure out how to code the sequence that I wanted to code into DNA. But I just found already existing template in the internet that I used. So I wanted to translate my name and surname into DNA language, and then think maybe in the future for how I can incorporate a signing technique, like in signing your art with this signature, but not only artworks, but maybe also we have a discussion during the exhibition last year that with Günter Seyfried, who is here on the call, that in Austrian Cultural Center, people were asking, can you sign, for example, your babies with your DNA signature, which is already actually signed in the babies because you're giving them your DNA, but additionally you can just insert this this tag. So that was a funny question. And another layer of this project that was related to finding the similar organism. So after having my name and surname translated into DNA sequence, I was looking in this web page with this whole bibliothèque of genome. So I found organism who is has 89 similarity with has the same sequence of letters, and that's the marine bacteria Schwannella, which is anaerobic and is a deep sea bacteria and has some interesting capacities that I haven't checked yet because I didn't have a chance to acknowledge this bacteria, but that's another branch of the project that I can go back to and have some kind of relationship with this organism. And
can I take one more step to what you did, because I think it took me a while to get it. But so what you did is you use this table, right, which takes our alphabet. Yes. And every letter in the alphabet stands for amino acid. So you took your name.
Exactly.
So that would be
and I created my name from
AG.
This based on this code.
Yeah.
And then it's actually synthesized, but the project stopped there at this moment, and maybe it will be revitalized at some point.
Oh, this is the synthesis. So this is your name in the
yeah, exactly. And then I made then I put it to, I don't remember the name of this program, but there is like this tool that enables you to visualize DNA. So just like put the code there and you have this visualized. And that's the image of Schwannella, the right top bottom, the right bottom. Yeah, it looks pretty nasty and dark.
How did you spell your name and DNA, actually?
How did I spell my name? Oh, if Roland will go back. So for example, you see like you have K and that would be listen. So it would be AAG. So I did like every letter was like have the amino acid signed to it.
Okay, I didn't have it on full screen. Now I see it. Thanks. Cool idea.
So that was a very simple coding, and I was like inspired by Joe Davis, who is a pioneer in like this kind of idea of coding into DNA information, which is not producing any proteins or.
Mm-hmm. And did you think about like extracting some of your own DNA and sequencing a part of it, so you can like sign with actually your genome and not just with your name? You know, like it's like metaphysically speaking, it's like more you than your name.
That's true. No, I didn't think about it. I think I was also referring to this art tradition of signing the paintings with your name and surname. That's like my initial background. So, and that was like some kind of also like making a conversation with this this urge to have your work signed, the value of the signature, and the place of this in art market, which has like an art milieu, which has a very different dynamics than the biological milieu. So if I sign the, let's say, bacteria with my name and surname, and it's being copied, and I have like thousands of copies of my signed organism, like this is like the opposite of what is meant in arts, where you have to have a one unique piece that is very carefully cherished. And so that was like another like
you wanted to give the traditional approach longer or a bridge to the bio art tradition that develops, right? Like because when you, for example, when you would take like some part of your genome that's just specifically for you, it stands for you. So it's like a very personal thing to share and to sign your work with, but it's not touching the traditional of artists before. You know what I mean? It's like a totally
I think it's a great idea. Like that's a good extension of thinking in this direction. But I didn't do it. I didn't do it. I didn't think about it at that time. But that's very cool, Catherine idea. I like it. You should do this. I will sign my babies with my genetic code if I ever have any.
You can tattoo it. It grows with them.
You are getting freaky here. I want to pull out some things people wrote in the chat. Maybe Patrick, maybe you can unmute and kind of share what you said, because it's really cool about the blasting.
Yeah, so the link that I posted there is to a program called BLAST, which is how you find similar sequences in DNA or in protein sequences. Right, so we're looking at amino acid sequences here, which are 20 letters out of the 26. So you can just type your name in there, and then at the bottom you scroll down to the button that says BLAST, and it'll search all of the protein databases and find as good a fit as it can for the name that you typed in. So I have a short name, so it comes up with a whole list of different proteins that contain my name as an amino acid sequence.
Cool. And then there's two other comments which are nice to share. Judy McGuyan says, embed your DNA in your art and make sure everyone knows its provenance. I don't know what provenance means.
Where it comes from.
Where it comes from. All right. Everyone knows where it comes from. And Anders Larsson Tevis says, this also reminds of a Bach signature cadence, which is also nice. It is a way. It is.
Oh yeah, Bach. It's very cool about Bach and very similar. Okay. I'm also reading chat right now.
Is there someone else who wants to do kind of a last question and answer with Carolina?
I have a question for Carolina.
Yeah.
You said the Schwannella is the closest bacteria that you found to the sequence. I was wondering if you feel any connection to that species, if you feel like it represents you well, or if it's somehow like you.
Immediately, it's my spirit animal, and my friend Stanisław, since then he's calling me Schwannella. So that became kind of my nickname. Deep, marine, like anaerobic, like I wish it would be great to encounter Schwannella at some point and like collaborate with this species. I would love to do that. So that's like on my bucket list.
This is amazing, but this is something we all can find out now, right? With this BLAST instruction, we can all find our kind of soul mate
spirit animal
signature spirit thing. Yeah.
Oh, that could be like a great workshop, like working, like finding your spirit animal, like a spirit bacteria, and then like figuring out what kind of collaboration you can make with this species.
Would be amazing. Thanks,
I'm
Carolina.
noting it down. Thank you.
I think we have to move on. Let's give a big applause. You can also unmute if you want to. Or you can applause, still muted. Okay. We see you. We see you. Then there is more later, but we have Günter Seifi joining us as well. Carla Mullins was going to join us, but she's delayed in a work meeting, which can happen if you live on all kind of different parts of the world. But I also wanted to bring out my personal project because I never really showed it. It's kind of fresh. And so I'll present that for 5 minutes. I show the thing I uploaded, and then we go to Günter, which is, Günter's here, right? Günter, can you say something?
Yes, hi, I'm here.
Yes, okay. And then we go to Günter. Hey, Günter, which is Mutants from Inner Space Revisited, which has also some echoes of Carolina's project. Now, I wanted to present shortly this project, and would be curious if you have more questions. So this is the bigger project is called Tele-Agriculture, and it's kind of spearheaded by Julian Staden, who couldn't make this tour today, but he is at the BioSummit. And actually the first idea for the project Tele-Agriculture was invented at the BioSummit. So this is also an encouragement to mingle a lot and chat with people and get great ideas that maybe 2 years later can be presented at the BioSummit. But the idea is that it's a project combining aquaponics. You know what aquaponics are? No, maybe I shall explain it. So aquaponics is when you grow fish and plants, and kind of the fish poop, and that feeds the plants, and then the plants feed the fish again. So you can make this kind of, oh, I should show some pictures as well. It's here is a system. So here is the plants, and then below there is the bucket with fish. And that's an automated system in this case. So we try to automate it and to put all kind of sensors in there which kind of live stream this data, and then this data and as well as chemical sensors of the temperature and all other kind of factors in this system can be used by biologists to interpret, but also by artists to make installations with it. And that is the idea. It's kind of a community project. You can visit it at dailyagriculture.org. And the idea is that with this modular aquaponics kit with the plants and the fish, you can make it differently in different situations and kind of adapt it to your own environment. And this was in Portugal at Cultivamos Cultura, which is a bioart laboratory in the south of Portugal. And these were two ecosystems, one freshwater ecosystem and a saltwater ecosystem. By the way, my chat somehow disappeared again. So if somebody has questions, just yell. Maybe I can open it again. Here, chat. But yeah, so this is me. And what I did is this residency in Portugal, and I looked a lot at kind of the health of the ecosystem and what we can actually do with intermittent testing using the microscope to see what kind of organisms are in there and analyze that. Here's crabs as well. But what I mainly did as part of this project, oh, I didn't know you put that photo on there, is document the whole process working there with Julian and kind of thinking about what the concept of building such an aquaponics kit is and kind of growing your own food. And we had a late night conversation about this, about that there is no such thing as peripheral skills. So the project is called No Peripheral Skills. Like every skill you have in life, I mean, we started with making fire and that was essential, right? But now you can kind of order your food via Uber Eats and we kind of lose these skills because we can outsource everything. This was a bit of a late night conversation, I have to say. But anyway, we recorded it and I used that audio in a movie kind of documenting what we were doing, which you can see here later. I won't show it now because it takes, I actually don't remember how long it is. It's 4 minutes, but I don't want to show it now. But I made that movie kind of using that audio and showing microscopy footage and other footage of the crabs and kind of the atmosphere. And I'm very curious what somebody thinks about it because it's the first time I show this video. And then the second video is Julian makes a lot of glitch works where he takes data and uses the data stream to glitch up images. And so I made a glitched version of this video as well, where the audio and the video is kind of completely distorted. I can show a bit without sound. I can play them both without sound. So, so you have something to look at, although probably it will be really buggy. Okay, I suggest we move on soon to Günther, but does somebody have any questions about this? Julian is also there soon, and he wants to kind of extend this idea of having more tele-agriculture kits around the world, so people grow their own aquaponic systems. Would anybody be interested in growing them at the Counterculture Lab, for example?
Definitely.
Yeah,
Sure.
We have a experiment actually or project going on. How interchangeable is it?
Yeah, how interchangeable is it? So one of the things we're interested in is indeed kind of having people this kit, but they use their own local ecosystem. So in this case, we took fish from the lagune in Portugal, in Faro, and other fish we had from the fish shop. So these were freshwater fish, and the other was kind of brackish, kind of these crabs were also in there. So the idea is that if people live in different places around the world, they would make this system with different fish from their local environment. Anders Larsen David asks what sorts of microscopy techniques did you use for this project? So this is just light microscopy, yeah, just a standard light microscope, although it was kind of special because it had also the ability to shine light from from below, which was kind of diffuse instead of very centered. It was a beautiful microscope. And then on the right, actually, I used a different technique to glitch it up in processing to kind of keep the dark pixels or keep the light pixels, so you can create this glitchy videos. What food were you growing? Tomatoes were doing it quite good because they also like salt, and it was a salty system. Tomatoes, and we had this other plant which I forgot the name of, which is also, it's this kind of plant you have in fancy restaurants, on like salty plants. Salty, you know, what is kind of a seaweed, but it's called samphire. We had sapphire. Thanks, Nell. Sapphire we had. Well, thanks everybody. I think we're gonna continue because we only have 8 minutes left, and I want let Günther present as well. And so we're gonna go away from this exhibit and go back to the main hall, and go to Günther's work, which is Mutants from Inner Space Revisited.
Hello, everybody.
Yeah, maybe you can first kind of tell a little bit about the work, and then people can ask questions after.
Yes, this is just like old work revisited. So like in 2004, I started this project where we translated digital data into DNA, and we had different kind of softwares. And this was one of the second projects. Was we had a small GIF, an animated GIF, and we made in vitro experiments where we tried to set up conditions like you get like mutations from toxicity or nuclear radiation or those kind of things. So by changing the magnesium chloride level of the amplification. And then here we were invited for the Ars Electronica to join a project with Spela Petric, and together with Roland, it was called Semiotics of the Laboratory. And so it was like included in the bigger project of Spela, and so we had like little experiments at the open lab at the Ars Electronica Center. And so there we took, again, up the older year, we had a little picture translated into DNA. Can you show that? This one, yeah. So this was also nice for a software test. So I used the gradient to avoid repeats. And then we made PCR reactions and raised the magnesium chloride level and tested some other chemicals. And then you could see, you extract the DNA again, send it in for sequencing, and then you can see the mutations in the images. This is like mutation.
So you took this picture, you translated into DNA with some kind of algorithm, that every color is some DNA code. And then how come there's 3 different pictures with different colors there?
Yeah, you remember?
Yeah, but
we
I'm
were
also
doing some of them we did together. You know, we were like pipetting like the whole 3 days, and had like I don't know, like tons of different PCR runs. And those are the few that that worked actually.
But every picture is a different tube or a different sample.
Yeah, that's a different tube. Yeah.
What is each square in a picture?
No, it's called a pixel. Usually this is a zoom in.
Okay.
So this is like what do we have here, like 6 to 8 pixels or more. That's the size of the image. So the software is not like specialized on translating a lot of digital data into DNA. So it doesn't have any compression algorithms. It was just like a direct translation, so it's possible to see small changes. Because if you have like a compression algorithm and you change just 1 bit, then there's a totally random outcome then.
You have some more questions in the chat. How many bases are representing a pixel, Catherine asks?
12, yes.
12.
It's a 24-bit image.
Yeah.
And you have like 3 colors, RGB, and 4 bases. So that's how the algorithm works.
And
then there's
Günther, this would be awesome to make it into embroidery like an RSA code, which it definitely would.
I can share from the screen. I can can share the original work if you want.
No, I can only share the screen. That's the limitation here.
Yeah, never mind.
But people can see it.
I can send in the link. They can look for themselves.
Great. Are there any last questions for Günther?
Yeah, I also have one. What actually motivated you to go in a project like this? Like, was it the DNA as a new storage media movement in academia and industry, or was it something different?
No, actually not. It was like totally by accident that I came to this. Also, like that was like the entry point into bioart for me as well. So there was like, when I was studying at the university, there was one project where you can propose a project in the course of a travel with a ship from Vienna to the Black Sea on the river Danube.
Okay, Günther, I have to say we only have 1 minute left. I feel this is going to be...
but because that's a long story, yeah.
Yeah,
so,
I felt it coming.
In the course of those projects, it came to an idea to translate the movie, and I asked a friend of mine who was a biologist if this is possible, and he said yes, but you have to downsize it, and so I just came in by accident. And for me, like I did like a few experiments, it works more like a epistemological machine. Only in the last work, I really got to know what the actual problem was in this.
Okay, everyone. Günther, I know this is like a big thing, but we can talk hours about this. I would say let's continue these conversations on a Slack thread, which we start when this wraps up in the Bioart and Design channel. But what I do want to say is, first of all, thank you all really for joining this tour. I hope you enjoyed this way of seeing some projects on the gallery. And the second point is there will be more tours every day by different people, and then different projects will also be presented in, so you can join again. And I think Carolina, on Sunday you have an idea to do it with scientists who actually not present the work, but kind of reflect on their view on the work. Anyway, you can join every day and have a completely new experience. So thank you all for joining. We have to say goodbye now because the workshops team has to prepare the next round, I think.
You guys should do a group photo really quick before you go. Yes.
Thank you, David. You just popped in. Yes.
Yeah.
You guys are great. That's it. Got it. Awesome. Enjoy, everybody.
Yeah,
Enjoy
hopefully see you on Sunday and Saturday during the gallery tours. Bye.
Bye.
Bye, everybody. Thank you for coming.